So you might have heard the word CRISPR being thrown around in the media in the past few years, and if you're wondering what it is, it's a new and kinda revolutionary method of targeted genome editing that's been giving biomedical engineers one heck of a field day. It's been described by some scientists as potentially one of the most powerful forms of technology ever discovered by mankind. And the best part? Well, we didn't actually create it. CRISPR-Cas9 is a naturally occurring part of the defense system of bacterial cells against viruses. They basically serve to search and destroy viral DNA, and we're just harnessing this power. The story starts back in 2013, when researchers at AdGene first reported the use of CRISPR-Cas9 to selectively slice genomes in human cells. Since then, scientists have been going all out, creating the first monkeys with customized mutations as well as immunizing human cells against HIV. In 2015, Duke researchers were able to use CRISPR to successfully treat adult mice with Duchenne muscular dystrophy. Just this past year, the National Institute of Health approved clinical studies of CRISPR in the US, and the first case of using CRISPR gene editing in a human patient was conducted in China. But what is CRISPR-Cas9, and how does it work? Well, for starters, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and Cas9 is a CRISPR-associated enzyme that's programmed by small RNA to cleave DNA. Think of it like a pair of scissors with an attached GPS navigator. What CRISPR basically is a collection of sequences on DNA, and they're transcribed onto short RNA sequences, called CRISPR-RNA, that serve as kind of a search template for matching DNA sequences. When the target DNA sequence is located, Cas9 binds to the DNA and cuts the strand at that site, which effectively deactivates the gene. It's also possible to activate gene expression using modified forms of Cas9. Why is this cool? Well, scientists can basically use it as an on and off switch for certain genes to figure out their exact function in order to better understand how the human genome works. See, 98% of the human genome doesn't code for proteins, and while we don't quite know what it does, it's hard to believe that it doesn't have at least some important function. Now we can find out! So is that what all the fuss is about? Well, there's actually more. While genome editing has been around for a while, CRISPR allows scientists to edit genomes with unprecedented precision, efficiency, and flexibility. The human genome is 3 billion letters long, made of combinations of A's, C's, T's, and G's, and while most DNA slicing enzymes are programmed to cut whenever there's a short sequence of maybe TACG, say, Cas9 can recognize sequences around 20 bases long, with guiding RNA sequences that can be tailored for targeting specific DNA sites. Think of it as the search function you use when you look through a document. How many results are you going to get for typing in "the" versus if you type in something like therapeutic music? Another cool thing is that CRISPR-Cas9 can also be used to target multiple genes simultaneously, which makes it highly efficient and differentiates it from other forms of gene modification currently used today. It can also be used on practically any animal on the face of the earth, so that's a plus. The biggest thing is that it has huge applications in immunotherapy, and has the potential to cure genetic diseases like sickle cell anemia and Huntington's disease by searching for and correcting mutations in the genetic code. Pretty exciting, right? Well, that's all for now, and I encourage you to look for new developments in CRISPR technology in the future. Bye!